Hello guys, welcome back to my channel. Today I'll explain about a disease called Brucellosis or Brucella. What is Brucella? Brucella or contagious abortion is a bacterial infection found in livestock. The disease is transmitted from animal to animal, so it's a zoonotic bacteria. So it means Brucellosis causes abortion. We found something here that Brucellosis causes abortions at the late stage of pregnancy. As we said, it's a bacterial infection. Causes abortion. And it's contagious. And it's found in livestock. It's found in even dogs and cats and swines. Mostly in ruminants, and it's zoonotic, and it's transmitted through first drinking raw milk from an infected animal. So that it means if the milk is not pasteurized, you will get infected by the disease, by the bacteria. It's transmitted from animal to human through direct contact with discharge in the genital tract. So that means we find something else here. Brucella causes discharge in the genital tract. So it means it causes penis discharge in males and vaginal discharge in females, in female cows. We have a note here. It causes cattle infertility if mating occurs between male and female. If mating occurs between, or sexual intercourse, we can say, occurs between a male and female, that if a male is infected or a female, one of them is infected, it causes cattle infertility. Etiology. There are many species of Brucella, but the pathogenic species are, first, Brucella abortus, mostly responsible from cases in cattle or bovine, we can say, about bovine cows. It causes abortion. Brucella melanosis, mostly responsible from cases of sheep and goats. Brucella suis, found in swine. Brucella canis, it's found in dogs. Brucella ovis, it's found causes epidemitis of rams. But the three most important of them are Brucella abortus, Brucella melanosis, and Brucella suis. They are the most main important types of Brucella. Now we go to diagnose. How is Brucella diagnosed? We have three main ways of diagnosing Brucella or detecting Brucella. First, sending fetus to the lab to isolate the bacteria. First, a fetus is sent to a lab to isolate the organism or bacteria in a cool box, in a cold condition, and stomach contents are taken and inoculated on special media. And after that, it's incubated, of course, and we see the results. Second, we have culturing. How it's cultured. We have two media. We have one selective media, and we have one general media. The general media is culturing sample on McConkey agar. I write one of the C capital to make it easier to pronounce or read, to not get confused between them. Or a specific agar, it's called Brucella agar. So we have two main agars. We have McConkey and Brucella. Why we use McConkey? Because McConkey agar is a specific agar or media that's used just for gram-negative bacteria. For example, Pasteurella, for example, Brucella are just gram-negative bacteria. And it's incubated in the temperature of 37 Celsius at two conditions. The two conditions are aerophilic or microaerophilic. And the bacteria is aerobic, we know. And after growing of colonies, a bacterial smear is done and fixed. After fixing, a gram staining is done, of course, not just fixing or smearing. A smearing is done and gram staining, and it's fixed. After fixing, it is observed under the microscope by using the oil immersion. Yeah. Third, we use antibody test called serum, rose Bengal test. The serum is separated by centrifugation, by centrifuge machine, and its rose Bengal reagent is added. If agglutination occurs, it's positive. So here we have gram stain of Brucella. We have Coccobacilli of Brucella. It's gram negative, as you see. They are so small at size. And we have the third type of test, as I said, is the rose Bengal test. How is it done? Serum is taken and antigen is added to the serum, called antibody, called rose Bengal reagent. If agglutination occurs, the test is positive. Here we have serum is added on each wheel of those, and after that we add rose Bengal reagent on both. As we see in the number 9 and 10, those two wheels, number 9 is control. Number 10, we have agglutination. That's a positive reaction. You see, that's a positive reaction. But in number 4 and 5, number 4 is control. Number 5, no agglutination occurs. It is negative test. We know that we have vaccines for Brucella. I will explain the vaccines that are used for Brucella. For one of the controlling ways are using vaccines are important to protect against brucellosis. And brucellosis comes back every year. For example, if a cow is treated with brucellosis, if a cow is treated, and after a year the cow will get reinfected by brucellosis. That's the problem. Not like Campylobacter, Campylobacteriosis, ovine genital Campylobacteriosis. No, it will not come back again. But brucellosis will come back again. First, the vaccines that we have. Strain 19 used mainly for calfhood, and it's not recommended for males. So it means the vaccine is just for females. We have 45/20 vaccine, or killed Brucella. We can call it killed Brucella. It's used for calfhood and it's used for Brucella abortus. Which type of Brucella is it used for? It's called killed Brucella, or 45/20 vaccine, and it's used just for Brucella abortus. We have Reps 1 vaccine. It's used for lambs and kids. It's used for in cases of Brucella melanosis. And we have 53H38. It's used for lambs and kids. So, thank you guys. Hope you get benefit.